Welcome everyone. Thank you for joining us for today's presentation. My name is Elizabeth Firth and I'm the Senior Product Manager of Human Genetics at DNA Genotech. DNA Genotech is the leading provider of products for biological sample collection, stabilization, and preparation. Today you're going to hear from Dr. Yen Wang about her work in salivary epigenetics. She completed her PhD in biotechnology at Deakin University and is a postdoctoral research fellow in the Early Life Epigenetics group led by Professor Jeffrey Craig in the Institute for Mental and Physical Health and Clinical Translation. Her research areas involve investigating and developing epigenetic biomarkers for neurodevelopmental and other chronic disorders. Currently, she's leading a project on optimizing saliva cell type separation for the early identification of hemolysis in leukemia patients. She's working closely with the School of Psychology and the Institute for Mental and Physical Health and Clinical Translation at Deakin University, as well as in collaboration with the MCRI and Royal Children's Hospital. During today's session, Dr. Wang will share a study investigating sample collection methods and cellular heterogeneity using cytology compared with cellular deconvolution algorithms. The title of her talk is A Comparison of Epithelial Cell Content of Oral Samples, Estimated Using Cytology and DNA Methylation. Now, without further ado, I'll turn the floor over to Dr. Wang.
Firstly, I would like to thank for giving me this opportunity to present my research. It's a pleasure to be here. My name is Yen Wang, and the title of my research is A Comparison of Epithelial Cell Content of Oral Samples, Estimated Using Cytology and DNA Methylations. In this slide, I would like to give a brief introduction about epigenetics and DNA methylations. Epigenetics refer to stable heritable traits that can't be explained by change to the DNA sequence. Epi, a Greek prefix, means on top of genetics. So it refers to another layer of control on top of the DNA sequence. So epigenetic is the regulation of gene expression through mechanisms that do not alter the core nucleotide sequence, which a change in phenotype without a change in genotype, and in turn affects how cells read the genes. Epigenetic change is a regular and natural occurrence, but can also be influenced by several factors, including age, the environment or lifestyle, and disease state. DNA methylation is one of the mechanisms, a chemical modifications of the cytosine C base through the addition of this methyl group. This primarily occurs on Cs in the context of CG dinucleotides, which we call CPGs. When we study methylations, we are usually interested in finding methylation difference because methylation is critical in determining cell types. We often want to know what the differences between cell types are. Different cell types have different epigenomes, even between different types of blood cells. So, epigenome-wide association study, or EWAS, are particularly sensitive to unknown confounders. So EWAS involve of comparing lots of cases to lots of controls to find some difference in methylations. EWAS are often looking for small effects, such as those that might be present in complex disease or due to environment exposure. Cellular heterogeneity is a major confounder of EWAS due to the cell type specific state of DNA methylations. This is particularly the case in oral samples, which are a mixture of epithelial cells from the ectoderm germ cell lineage and immune cells from mesoderm lineage. As shown in the image here, cellular heterogeneity is important to suppress inflammations and false positive. In our previous study, and others have found that cellular heterogeneity in oral samples is influenced by the methods of sample collections, which buccal swabs containing much higher proportion epithelial cell compared to saliva. And we have also shown that epithelial cell proportion is also strongly influenced by age, Here the image shows children and adults, as you can see, influenced by age and oral health status as well. As you can see, the gingivitis in saliva. So now, how do we know cellular heterogeneity is a major confounder in your data? So by deconvolutions of cellular heterogeneity can be achieved by measuring the proportion of each cell type using cytology of collected cells or through algorithms that use DNA methylation data from specific cell types to generate estimates. Such measure can then be used in EVAS models to correct for cellular heterogeneity. So the image shown here on left-hand side is an example of how the result look like. So the y-axis is the cell fractions, x-axis is the case and control, which the n is a negative control, and different cell types, which include epithelial, fibroblasts, and immune cells. So our rationale for the study, buccal and saliva samples have proven utility for epigenomics and other cell-based omics, as these samples are a mixture of epithelial and immune cells. Therefore, the convolution of this cellular mixture is of utmost importance. Although cellular heterogeneity, although cellular deconvolution algorithm based on reference sample types have been applied to epigenomic study, but to our knowledge, the validity of such algorithm has not yet been tested using cytology of primary samples. Therefore, we aim to address this issue. So far, there are two studies comparing the cytology estimate of tumor purity to DNA methylation-based estimate and mRNA expression-based. So these are the two example studies. So our first aim is to compare epithelial cell content of buccal samples via oral collect DNA kits and saliva obtained via passive through collected in orogene DNA kits. Our second aim is to use customized DNA, oral collect DNA collection instructions. We compare two similar methods of collection differing in collection site and durations. Both of these aims will perform to measure using cytology and estimated with the reference-based epitish algorithm. So we hypothesize that the estimations of epithelial cell content would be highly correlated between the two methods, and the two similar methods of collection would correlate with epithelial cell content and DNA yield. So for the methods, there are 20 adult participants in this study. Their age range is 21 to 48 years old. The participant also completed an oral health questionnaire, which include questions about mouth injuries, oral infections, medications, and smoking status. Oral collect DNA is a sponge tip oral samples collection kit to mechanically rub off cheek cells, which is the OCA and OCB. So both of these two are oral collect DNA kit, while for orogene DNA is a saliva sample collection kit. So 10 minutes prior to sample collections, they were asked to rinse their mouth with water before cheek cells and saliva sample collections. So the main difference between OCA and OCB is the time spent rubbing the cheek. For oral collect DNA, participants will gently rub the sponge 10 times in back and forward motions in the furrow between their lower teeth and inner cheek on one side of their mouth. While oral collect DNA B, the sponge was rubbed up and down against the inside of the cheek 20 times and then rubbed 10 seconds in back and forward motion in the furrow between the upper and lower right teeth in inner cheek on opposite side of their mouth. compared with saliva in which no cheek rubbing is performed, which they were collected unstimulated via passive draw for 3 to 5 minutes to allow sufficient time to collect 2 ml of the saliva samples. After the collections, the samples was smeared on slide, and the slides were stained using diff quick as detailed elsewhere. All slides were de-identified and analyzed by 2 observers. Counts were used if the discrepancy between observers was less than 10% of the total count of the count for each cell type. So a minimum of 50 epithelial cells and 100 cells total was counted. Cells were scored as epithelial cells or immune cells, the latter including segmented cells, lymphocyte and monocytes as shown in this picture. So this image is looked under the microscope for in 100 times magnifications. After that, the genomic DNA the saliva was genomic DNA extracted following the manufacturer's protocol, and DNA quality was measured using a tap stations. Following genomic DNA extractions from all the samples, these DNA samples were treated with bisulfite to convert unmethylated cytosine into uracils and transformation of uracil into thymine by amplifications. So genome-wide analysis of DNA methylations was assessed using infineal methylation epic array with probes of over 850,000 methylation sites. So this is an example of the chip. So one chip you can put 8 samples. So these sites are for human only and have been carefully selected to be in and around genes and regulatory regions. There are 2 probes for each CpG. So one for the methylated version and the other for non-methylated version. Data was then normalized within array and between array. The epidish and RPC algorithms were applied to estimate the proportion of epithelial, fibroblasts, and immune cells from methylation epic array data. So the first result shown here is the epithelial cell proportion determined by cytology and DNA methylations. The x-axis represent the different collection methods, which is the OCA, OCB, and OCG and OG, while the y-axis represent the percentage of epithelial cells. The lighter color boxes represent the using cytology to measure the percentage of epithelial cell, while the gray color boxes are estimated with reference-based epidish, which we here indicated as DNA methylation. Firstly, for the cytology result here, the lighter boxes, we can see that the mean proportion of epithelial cell in saliva was significantly lower than with the sponge collection methods, which is OCA and OCB. So you can clearly see there was a 28.5% mean difference between saliva and cheek swab methods. However, for DNA methylations, we see a similar pattern. So we found that the proportion of fibroblasts was negligible, and immune cells and epithelial cells are highly negatively correlated. The mean proportion of buccal epithelial cell determined by DNA methylations in saliva was significantly lower than cheek swab methods, which is similar pattern as the cytology methods. So for both estimation methods, the cytology and DNA methylations, there was no evidence for a difference in epithelial cell proportion between these 2 OCA and OCB method collections. There was also no evidence of an influence of recent gum bleeding and sex on epithelial cell proportions across all methods of sampling. So next, we did an analysis on DNA yield. So the x-axis represent collection methods for different collection methods, and y-axis represent the DNA yield per 1 ml in microgram. Saliva samples show a significantly higher mean of total DNA yield per ml, which is around 8.2 times higher compared to oral sponge collection methods, which is both OCA and OCB. Although the DNA yield was approximately 1.7 times higher in OCB, slightly higher compared to OCA, the sponge method, this difference was not significantly different. So next, we also look into the cytology versus DNA methylations. So x-axis is the percentage from cytology, while the y-axis is the percentage from DNA methylations. So this result is to comparing the methods of estimating cellular heterogeneity in oral samples to address our hypothesis that proportion of epithelial cell present in oral sample estimated using DNA methylation analysis represented the cell proportion as measured by cytology. So we pull all samples and compare both methods. The two methods were strongly correlated. As you can see, it's 0.84, highly correlated. However, as you can see, the intercept of the line of best fit on the x-axis was 34%, which a 20% mean difference of DNA methylations compared to the cytology methods. So both methods are correlated similarly in buccal and saliva as well. So next, we also look at the age effect on epithelial cell content. So in our previous paper, TIDA, we observed that epithelial cell content of buccal sample of buccal swabs and saliva was lower in adults compared to children. To investigate a possible age effects using epithelial cell content estimated using DNA methylation, we combine both buccal swab and saliva data from this study with 7 of our other infinium array study, including 3 published and 5 unpublished. So the image A here on the left-hand side is comparing age versus percentage of epithelial cells for 8 of our array study, including this study. So each of the different study are indicated by different colors, while the B image is comparing the age, is comparing the percentage of epithelial cells by study. And the bracket below of each study is the mean age for the study from young age to senior, as you can see here. So both, we found a moderate negative non-linear correlations between age and the epithelial cell content estimated by DNA methylations. So furthermore, we stratify buccal and saliva and look at them separately. So this result, we analyze the buccal, which is the A graph, and B image, and B graph here is the saliva. So we found a similar relationship, which is moderate negative nonlinear correlated comparing age versus percentage of epithelial cell from buccal and from saliva. So as summary, our findings suggest that the type of buccal collector has minimal influence on the proportion of epithelial cells collected. And this may also be one reason why increasing collection time for oral collect sponge did not increase the proportion of epithelial cell collected, nor did it significantly increase DNA yield. However, future larger studies are needed to further test our hypothesis. However, there may be a danger that longer collection time may penetrate blood capillaries within the inner cheek, which would increase the proportion of immune cell and may negate any rise in epithelial cell numbers. And in our present study, our expanded data analysis show that children and adults show a large within populations variations in cellular heterogeneity, and the epithelial cell content of saliva, but not buccal samples, was significantly higher than in our earlier study. So these agree with our previous finding that age has a much greater effect on saliva than on buccal samples. We also found that the epithelial content of oral collect DNA collected samples was around 47% higher than of saliva. So this difference was 11% larger than that of our previous study, so which may also reflect an age effect. So our result validate the use of epigenetics algorithm in estimating the effect of cellular heterogeneity in EVAs. Our line of best fit showed that DNA methylation underestimated the epithelial cell content determined using cytology by 34% at 0% epithelial cells and by 30% at 87% epithelial cells. This is larger than the over or underestimation of up to 10% observed in the previous study of blood. So this discrepancy could be for a number of reasons. The reference data set was derived from Illumina Infinium 450 array data from 11 different epithelial cell lines, which may not accurately represent buccal epithelial cells. We also cannot rule out the possibility that buccal and immune cell types may have been differently applied to slide prior to cytological examinations. Nevertheless, the high correlations between epithelial cell proportions based on cytology and DNA methylation should still be sufficient to use the latter to generate estimation across a set of biosamples for adjustment within EVAs. And we suggest that both cytology and epidemic estimating methods are both reliable to use. So to conclusions, to our knowledge, this study is first to analyze the correlations between cell proportion in our samples, estimated using cytology and DNA methylations. Another strength in our longitudinal analysis showing a decline of epithelial content of buccal swabs and saliva with age. However, our sample size is relatively small, although our sample size for the study of age effects was much larger because we have 8 of the study sets. So for future study, we suggest that a larger scale study that compare estimate of cell proportion using both cytology and DNA methylation are required to validate our findings. So finally, I would like to acknowledge and thanks to my supervisor, Professor Jeffrey Craig, for his guidance and advice, and to our early life epigenetic group member as well for their support. I would also like to thank DNA Genotech for their funding, and Dr. Rafael, Dr. Mike, and Elizabeth for their advice and support. And I would like to thank Professor Lawrence and Dr. Tim for providing data, and Professor Andrew for his advice on the epidemic algorithms. Thank you.
Thank you, Dr. Wong, for sharing your expertise, and thanks to all of you for joining us. A replay of this webinar will be sent to attendees in the coming weeks, but it can also be found on DNA Genotech's partner page and on ASHG's on-demand content. If you would like to learn more or speak with DNA Genotech about our products and how they could help with your research, there are a number of ways to connect with us. For the duration of ASHG, you can visit DNA Genotech's partner page, where we'll have representatives available for discussion during the allocated times each day. Alternatively, we can be reached anytime at info@dnagenotech.com. And if you wish to trial any of the DNA Genotech products, You can also request free samples at try.dnagenotech.com/ASHG. I thank you for your attention, be well, and enjoy the rest of the conference.